Hi, I am Tori. Thank you all for coming. I appreciate you all being here and tuning in. So I'm going to take a little while to tell you a little bit about myself. I'm a research scientist here at NASA Ames Research Center, same place that Sanjoy works. So I'll tell you a little bit about how I got here and the path that led me here, and then what it is that I do here at Ames. So I am a chemist by background. I got my degree in chemistry at University of North Carolina a long time ago in the distant past. But early on in my time there, I started working in a lab that did marine sediment chemistry. And a lot of the chemistry there is mediated by microbiology. So that kind of gave me an interest in working in microbial systems. I think a lot of people in my field probably think of me as a microbiologist, despite having never had a single course in microbiology. Don't tell any of the people in my field that, please. So the work in marine sediments and in microbial systems really kind of carried me into NASA Ames. I took a postdoc here back in the late 90s and then continued into a permanent job here. And I've been at Ames for about 15 years now. And a lot of people at the time sort of thought, you know, what's the connection between working on marine sediments, mud basically, and NASA? And the connection really is that here at NASA, a lot of my work is involved in, trying to connect environment to biology. So to figure out what it is about environments that makes them habitable for life, and to figure out what it is that life does that changes the environment in detectable ways. And in both cases, there's a lot of emphasis on microbiology. And so my own work in understanding the microbiology of marine sediments, in what controls the distribution of life there, in what sorts of chemical signatures the life creates, really transitioned well to NASA. And it, for me, played to a deep personal interest in all things having to do with space. I was very much a spacey kind of kid growing up. I never had it as a career goal to work for NASA. It just kind of worked out that way. And I didn't even come here specifically to be at NASA. I came here to work with specific people and on specific projects, and it just turned out well for me. But the background in chemistry really has kind of stuck with me in terms of the way that I view biological systems. So I see them very much through a chemical lens, and in particular I see them through the lens of energy flow. So the one thing that is, I think, inarguably universal to life everywhere is that life requires energy. Everything that we understand about chemistry and physics makes that so. And making that statement is kind of meaningless in a sense, because energy is everywhere. It's the details of it that matter. How does life require energy? In what forms and how much? And that really has become the way that I study the world around me. So when I take a look at the microbial systems that I study here on Earth, and that's really how I pursue my research. I study systems here on Earth. I try to extract the general principles about them, the ones that are not specific to the kind of life under study, but that are general and transferable to life elsewhere. So when I study those things, really it's through the lens of energy availability. And all of the diverse projects that I have, the diverse array of places that I work in, all of them can be seen in one way or another in how life affects energy flow, or how life relies on energy flow. So a couple of examples are that we have some studies that we do here in Northern California. There is a site where we're studying the life deep underground. We've actually sunk some wells into the ground. And we here on Earth have a very kind of skewed view about biology, because we live in a world where biology is possible at the surface, where sunlight is available as a very abundant energy source. And so our entire context for thinking of life really has very much to do with places where energy is abundant. But if you go beyond Earth, if your interest, for example, is in whether life could be possible on Mars in the present day, or on Europa or Enceladus in the present day, really you're interested in understanding what life can do below ground. And the picture there is a very different one. So life moves at a much, much slower pace, perhaps hundreds of thousands to millions of times faster, slower rather, in terms of metabolism. And that's a very different context, one that is not well informed by everything we understand about microbiology. So when we study organisms in the lab, we specifically choose the ones that grow the fastest, that metabolize the most rapidly, because they're easy to study and they grow quickly. what we learn from them is not necessarily very transferable to life below ground, and yet it's those principles, understanding how life moves at a slow pace, how little energy is possible for life to get by. That's what will best inform our understanding about the possibilities for life on Mars or Europa. At the other end of the spectrum, I also have very much of an interest in exoplanets, and I think this is an extremely interesting and rapidly growing area right now. We know when I got here to Ames in 1998, I think we knew of maybe 2 exoplanets, something like that, and now there are thousands of confirmed or suspected exoplanets. There's quite a zoo of planets out there, and someday we're going to actually search for life on them. And what we're looking for there is not life that's buried deep underground. It's life that is so robust and so diverse and interesting, so capable and so productive that we'd be able to see it from many light years away just through its impact on its atmosphere. So we also have studies going on where we're interested in that, and there, my interest is in understanding how life mediates the flow of energy around it. So how is it that it takes, for example, on Earth, the energy of the sun and puts that energy into creating oxygen out of water, into channeling so much energy that we couldn't possibly imagine non-life to do it. So it's those sorts of signatures of life that become the interest where exoplanets are concerned. And a lot of this interest has carried me to places that I didn't suspect I would be able to go with a degree in chemistry and an interest in microbiology. So it's led to an opportunity to work a little bit on the design of some missions. So for example, there's a hope that there will be a mission to Europa sometime before too long, where there's a possibility to investigate the factors that may support habitability of Europa. And when they're doing that, when these missions are being put together, there's an interest in capturing a diversity of perspectives, including biological perspectives. And so I've had the opportunity to be involved in thinking about how you would design a mission and in speccing out how the big questions like how does energy become supplied in a place like Europa, how that translates into specific objectives and then even measurements and the details of those measurements. And that's all been just amazing for me, you know, for a kid who had a deep interest in space but never really thought about that as a career, to now be in a position where I get to be in on the ground floor of the design of missions like this has been kind of a remarkable journey, and you know, one that I feel fortunate about every day. So maybe that's enough of a sort of monologue. I think it's probably time for a bit of a dialogue and to see if there are any questions out there. Does anybody have any questions? Aha, there's one. Could you please tell us about the astronaut in the photo behind you? So this is actually not my office. This is Sanjoy's office. It was easier for me to come down and sit here where the software is already loaded on the computer. So I would have to get Sanjoy to tell you who the astronaut is. Sanjoy? Who is it?
It's Yvonne Kegel. The camera. Hi. This is Yvonne Kegel. She's an astronaut resident here at Ames. She's an expert in space medicine, and she had a bunch of questions about climate change that I helped her answer, so she gave me a signed poster of herself as a gift.
Very nice. Would you like a signed poster of me?
I see you enough every day.
Answer carefully. Okay, so a question from Sarah, more up my alley. What are your thoughts on the connection between information and energy? I think that if I were to come up with a statement about what a biosignature is, you know, I would say that life, in order to do things that are lifelike, in order to catalyze processes at many orders of magnitude faster than they would happen abiotically, in order to do things like create and replicate information, I think that I think that life has to have a unique capability to invest energy. So there are plenty of processes where you can imagine very large amounts of energy being put into processes, you know, high energy photochemistry and things like that. It's life's capability to parse out energy in small chunks as needed again and again. And for me, there is no more central place for that to be necessary in life than in the creation and replication of information. So life invests little bits of energy, but ultimately adding up to very large amounts of energy to correctly, accurately replicate information. And I think that's incredibly important. I think it's a way that we're going to be able to recognize molecules that are information rich as being uniquely biological. You know, do they stand out relative to what's possible biologically? So I think there's a very deep connection between information and energy. I think it's for me something that I've always sort of been pursuing and never quite able to quantify, but I feel like we're beginning to get there. And I hope that's enough of an answer. So next question from Adam Smith. Can you explain what your role is in your work with the Curiosity Rover? So I'm specifically on one of the instrument teams. There are 10 instruments aboard Curiosity. One of them is the CheMin instrument. CheMin does quantitative mineralogy by X-ray diffraction. So basically, you can pick up a rock and this instrument will tell you what is the specific mineral composition of the rock, what things are in it, and in what abundance. And the reason that's important is because the job of Curiosity is really to look for evidence of habitability in environments there, or past evidence of habitability. And the only evidence that we have available to us right now is the evidence that's present in the rock record. So you can imagine doing the same kind of thought experiment on Earth. You want to make a statement about whether a specific environment was habitable 3.5 billion years ago. And all of the things that made it habitable have gone, and all that you're left with are the rocks that either formed there or were processed there in the context of that habitable environment. So really, the job of CheMin is to say, okay, here are the rocks, what minerals are they made of? And every mineral, every rock, has a story to tell about the conditions of its formation. So some of them can only form in the presence of liquid water. Some of them don't exist above a certain temperature. Some of them don't exist below a certain temperature. You can imagine baking soda, which is kind of like a mineral. Baking soda ceases to exist if the pH gets too low, you know, if you sprinkle some vinegar on it or so. And so the more of these different minerals you can stack together, the more physical constraints that you place on your environment. So the job of CheMin is to tell us something about the mineralogy of the rocks we're looking at, and my job as part of the CheMin team is to then interpret that information and say what it means about habitability. Okay, so I'm looking at the next question from Sarah. So like many of us, you seem to have stumbled on astrobiology. What's your advice for people who are starting out knowing they want to do astrobiology? That's a really important question, I think, because astrobiology, you know, there's such a great diversity of topics in astrobiology, such a range of sort of interesting things to draw you in, that, you know, there's a temptation, I think, to just sort of jump in and do a little bit of everything. I think that what I would say is really important is to give yourself a strength in one specific area first. So, and when I say that, I mean, you know, be a physicist who does astrobiology, be a geologist who does astrobiology, because then you have that grounding. You know, in a traditional discipline, you have a detailed training that you can apply. And I came out of oceanography, so my undergrad degree was in chemistry. My graduate degree was in oceanography. And there's very much the same issue there. So you're talking about a natural system, the ocean, that is influenced by chemistry, biology, geology, physics, all of those things in concert. And it's a little bit the same. Oceanography has been around for much longer. And when you take oceanography in school, you don't just get a degree in oceanography, you tend to specialize. So typically oceanography is a graduate degree, although not exclusively. And you tend to specialize in one area or another. You're a geologist who then does geological oceanography, as an example. So I would say that's very important in astrobiology. Give yourself a firm grounding first. You know, allow yourself to understand the specific area, and then explore the field a little bit and figure out how to apply your expertise. All right, so another question from Adi. Could you please describe how you might experimentally detect life using the follow the energy idea? on Mars or Europa, for example. Oops, I've lost the screen. So I'm going to let Sanjoy come in and enter his own password back so that I can see your questions again. For example, is there diagnostic voltage resistance activation energy, delta G, for biotic reactions? So this refers to an idea that I've sort of been interested in for a long time, which is that if life rather is unique in its capability to apply energy, it may also be unique in its requirements for energy. So life's utilization of energy has two sides. One is that for an environment to be suitable for life, energy has to be available and delivered, you know, and in certain ways. So there needs to be a certain amount of power, a certain amount of energy available, and that's sort of, I think, what the question refers to. So that there are sort of discrete requirements for power for Gibbs energy in life. Whether or not those are then translated into unique signatures that we can see as a evidence of biology is unclear. So a good example is if you look at photosynthesis as it occurs around us. So life takes energy and invests that energy into splitting water. It takes the electrons out of water, the sort of H out of H2O, and we're left with oxygen. So that's a process that requires a very large amount of energy. It's something that life does very well and very efficiently, and yet it's also a process that happens all the time in the atmosphere when photons strike water. So there's a place where you might say, well, you know, life is channeling energy in a way that is very lifelike, and yet it's a process that we see happening all the time. And that's really the trap in thinking about what things represent biosignatures is, you know, life may do these things well, but, you know, if there's any possibility for it to happen in the absence of life, it ceases to be a viable biosignature. The way out may be to say, if life is capable of doing it so much faster than would happen in the absence of life, you know, so much more efficiently, then maybe there's a possibility there. And you could look at our own atmosphere, for example, as saying, you know, if you took all the oxygen in the atmosphere and all the reduced species in the atmosphere and you recombined them, how much energy is there to be had? And that sort of implies a certain amount of energy put in either by photons from the star or by biology that channels those photons from the star. You know, do you see something that really stands out? And if you did that calculation for Earth and for the other planets in the solar system, and that's been done by a guy named Dave Catling at University of Washington, you see that Earth really does stand out. It's really quite unique in how much energy is stored up in the atmosphere. And maybe it represents an amount that couldn't be done in the absence of life. But the other side to this, I think, goes back to Sarah's earlier question about the relationship between energy and information. So you can imagine, you know, abiotic processes that put very large amounts of energy into making something happen. So, for example, an X-ray or a cosmic ray delivers a huge amount of energy, but it delivers it all in one wallop and in not a very controlled way. On the other hand, if you took that same amount of energy and parsed it out into chunks, small bits, you might have a capability of replicating information. So when our sort of life replicates its genome, every single time it adds a new base pair, it invests some energy, about 50 or 60 kilojoules per mole, to make sure that's done correctly and more or less irreversibly. And when you think about the amount of information that is represented in a protein or in an entire genome, there's a huge amount of energy investment in making that happen. So I think if there's a way to specifically use energy as a very diagnostic way to indicate that life was active there, the information connection is the best one. But that requires that you actually see molecules that carry a significant amount of information. So that means they're large and heavy and unlikely to be found in an atmosphere. So those are the sorts of things we might look at or look for in places that we can physically access. When we're talking about something like an exoplanet, where the best we're going to get for a very long time is just the light from a telescope, then it's more the former. What sort of energetic signature is represented in the atmosphere? So let me read Sarah's next question and see if I covered it. Going back to the information-energy connection, you mentioned energy is important for replicators. You think all life will have something similar to genetic material that can be copied? So it's kind of a challenging question because it really depends what you mean by life. You know, I think sort of a working definition of life is something that seems enough like us, it's familiar enough to our way of thinking, that we allow it to be qualified as life. I personally think that one criteria is that life is about information. I think that energy flow is going to be universal to life. I also think that information content is going to be universal to life. Just because for something to be complex enough that we would recognize it as life, you know, that's something that doesn't just happen randomly. The chances of getting from one complex thing to a similar complex thing by complete random chance are extremely low. That's what energy is all about. It's making that improbable result happen reliably by investing energy into it. So yes, I think that anything we would think of as life will have something like a genetic molecule, whether it's a molecule like DNA, what Erwin Schrödinger called the aperiodic crystal, or some other form of storing information, you know, a crystal lattice with imperfections as an example, or any one of a number of different things. I think there has to be some means of storing information, but then extracting and replicating that information. Okay, a question from Enrica. Can you tell us more about your role in the Europa mission? What kind of life could be found there, and where within the ice shell versus the ocean? So it's important to say there is no Europa mission as yet. We're very hopeful that there will be one. You know, NASA and ESA and the other space agencies are consistently in the business of investing money into planning out and developing proof of concept for missions that may or may not happen. And the idea is that you begin with the big picture science questions. So in the case of Europa, there's very much an emphasis on understanding whether that place is habitable, whether life could be possible there. So how do you take that question and break it down into specific objectives that can be met with a spacecraft? And what measurements would you do in order to, you know, to address those objectives? And with what specificity and so on. And then you put all that in the hands of the very capable engineers, and they go away and they see if it's possible in the context of spacecraft design and what instruments are out there and so forth. So all of that process happens in the lead up to what might or might not be a mission. And that process has been happening for Europa during the last maybe 2 years or so is how long I've been involved. These sorts of science definition activities have been going on for Europa for much longer than that. And so my role really has been to try to make the habitability connection. So most of the people involved in defining the science of the mission have a much more detailed knowledge than I do about the instruments and about Europa and you know, the history of science there and what can be done there. I think where I come in is to try to specialize in saying, what does life really require when you think about it very generally, when you allow for the possibility that it could be different than the kind of life we have here? So what does life require, and how would you try to assess that requirement? So that's been really my role, is to bring habitability into the picture and then translate it into specific measurement objectives. So the second part of the question was what kind of life could be found there and where? Europa is a really interesting case. You know, when we think about astrobiology, there's very much an emphasis on microorganisms because they're simple, they occur earlier in the evolutionary process, they have a much broader range of capabilities in terms of metabolism, in terms of tolerance to diversity of environmental conditions. And so there's very much an emphasis on microbial life. I think maybe that's the most likely thing for us to find there. But Europa is interesting because there's a notion that life on Earth became very much more diverse, much larger, much more capable when there began to be a lot of oxygen in the picture. And what I think is not so much appreciated about Europa, there's a general notion that maybe there could be hydrothermal vents on the ocean floor of Europa. And so a lot of people tend to kind of put that picture in their head as what life might look like. When you think about the life around hydrothermal vents on Earth, there's a very reducing chemistry coming out of those vents, but it's coming into an oxidized ocean. And I think what's not so much appreciated about Europa is that there's actually a potential source of oxygen. So the surface ice of Europa is very strongly irradiated due to the magnetic field of Jupiter. And that has the effect of creating oxygen in the ice shell, at least we think so, and hydrogen peroxide. And those are things that could potentially give an ocean chemistry something like our own, a very oxidizing ocean chemistry. That's completely not known, of course. What is known is that there are oxidants in the crust. The question is whether there's a possibility of moving them, tens of kilometers down into the ocean. So there's so much that's unknown about what might be going on there. My own bias is that if we are to find life somewhere, it's most likely going to be if there is a point of intersection between the ocean and the rocky crust below. That's a place where energy is going to be made available, either by hydrothermal vents, if such things exist on Europa, or by the reaction of water with rocks down there. And then we would rely on the delivery of oxygen or other oxidants from above. A lot of unknowns about Europa, and I just really hope that we get the chance to go there and address those questions. Okay, so another question from Julio. I'm not an astrobiologist, so my question might be a little simple. What are organisms with the slowest metabolism do you know of, and how slow can Earth life go? That is a question that to me is extremely interesting. So as I said earlier, we have this bias about how life works by looking at the life around us, by thinking about how we function, how quickly we function, hummingbird-type metabolisms. There was some work that came out a few years ago from some of my friends in Denmark who went out to the North Pacific Gyre. So the oceans have a circulation that leaves the middle of them extremely poor in nutrients. They're thought of as ocean deserts. So there's very little sedimentation, very little nutrients. The water is incredibly clear. And there's very little delivery of organic matter, which is to say food, down to the sediments beneath. So they went to this place and they looked to the very bottom of the sediment core they were able to collect, which was about 80 million years worth of sedimentation. So at the bottom of that sediment core, the organic matter that had been hitting the seafloor was about 80 million years old. They still found microbiology that was capable of working there, but very, very slowly and at extremely low abundance. So relative to our own cellular metabolism, that life was functioning about 2 million times slower. So the average cell down there was metabolizing oxygen that was still present about 2 million times more slowly than would one of your own cells. And it's those forms of life, I think, that we need to be understanding if we want to understand the possibility for life in the subsurface of Mars or Europa. Okay, a non-scientific question from Sarah. What do I do for fun? I am very much an outdoors kind of person. So I grew up in the Bahamas. I think I could swim before I could walk, and I snorkeled on the reefs there all the time. And that has really stuck with me. And I got to spend my summers, a lot of my summers, with my grandparents in Canada. They had a place on a lake there. And I really developed a love for the outdoors there. And so almost everything that I enjoy doing, whether it's a sport or something else, has to do with the outdoors. I love to dive, I love to hike, I love to ski, any outdoor activity. So beyond that, I really enjoy traveling. I lived in a pretty diverse array of places when I was a kid, and I had a lot of opportunities to travel as a teenager and sort of in my early 20s, and that really has stuck with me. So I love traveling a lot because I think it really shapes your perspective. I think traveling is so valuable to your perspective, to see how other people live, to see the commonalities, what makes everyone the same, what makes everyone different, to sample a diversity of perspectives. And then lastly, I would say I love to cook. That's kind of my creative outlet when I have time on my hands, and that kind of plays into my love of travel as well, because I really love to cook ethnic food, try cooking things from all the different places that I've been. So those are all the things that I like to do, and what's really fun for me now is I have a son, Ryan, who's 8, and all the things that I have loved to do on my own for a long time, now all of a sudden those are beginning to be things that I can do with him. He's getting old enough that the outdoor stuff and the travel stuff, and that all makes an impact on him too. So it's really fun to experience those again. Okay, a question from Carol. Can you talk a little bit about serpentinization and the McLaughlin wells? So Carol's referring to a place that I and my group and a couple of other groups work at up in Northern California. And serpentinization is a process where very reducing rocks, kind of like the rocks that exist on our own Earth's mantle, are reacted with water. And when they do, they give off a lot of hydrogen. And this has gotten people very exciting because hydrogen is food for microorganisms. In fact, it's like rocket fuel for a diversity of microorganisms. So a lot of people have thought of this as a great place for microorganisms to live, or as a great source of energy for microorganisms. In particular, because that process doesn't happen just here on Earth; it happens on, presumably on the subsurface of Mars. We have actual mineral evidence for serpentinization there, and most likely in the subsurface of Europa and so forth. So what we do specifically at McLaughlin is we look at a place where this process is happening actively, and we ask the other side of the question, which is serpentinization not only creates hydrogen, this great food for microorganisms, but very, very alkaline conditions as well. And some of our wells in McLaughlin, so we've drilled down into the subsurface and looked at the chemistry of the fluids there, the microbiology there. So some of those wells have fluids at around pH 12.5, and that's almost as basic as bleach. So there's the great part to serpentinization. There's also the really tough part, which is how do you live in those very difficult circumstances. So a lot of what we're doing there, we have kind of a multidisciplinary team. There's my group that mostly focuses on the chemistry, and we try to constrain how much energy is made available by the process of serpentinization and how much it costs to live there in energetic terms. We also have a group that looks at the biology there, so they're looking at the makeup of the communities. We're trying to do some process rate measurements to see whether we can find evidence of metabolism that is suggested by the biological characterization that we do. And in the place that we work, so it's actually not very easy to find biological material there, perhaps not surprisingly. It's a challenging place to live. But we're beginning to construct a little bit of a picture about the ecology that lives there. And the challenge in a lot of these places, so there are a handful of places that are being studied for serpentinization, is that typically our access occurs at the surface. So we see a place where serpentinized fluids emerge onto the ocean floor or emerge into the atmosphere. And that's a very different circumstance. All of a sudden the chemistry changes by virtue of the addition of oxygen or of carbon dioxide or something that exists in that mixing area. And what we really want to do if we want to understand the possibilities for life in the subsurface is get down beneath that mixing zone and study the areas underneath. And that's where we're beginning to get in our project in McLaughlin. Okay, so there's a follow-up from Yasmin. I think along the same lines. What do you think about ocean serpentine rocks energy flow through terrestrial serpentine? So that's kind of an interesting question because a lot of the process of serpentinization is happening in the ocean. I think a lot of it is happening perhaps in ocean subduction zones. There are some places where faulting along the mid-ocean ridges has exposed the kinds of rocks that serpentinize. And when you find them at the surface, they're sort of isolated outcrops. So there are places where tectonic activity has scraped up bits of that ocean crust and exposed it on the surface. And there are significant differences. So the details get a little bit complex, but when you serpentinize with ocean water, it's a much different prospect than serpentinizing with seawater because ocean water is chock full of salts that change the nature of that serpentinization process. So a few things can happen. One is, I think that some of the work that Sanjoy is doing actually is showing that when you do the process with ocean water, you can still get out the same amount of hydrogen, but it really doesn't get quite as alkaline as it can at the surface. And that's an important effect because it means there's still plenty of food, but not quite as challenging a set of conditions. And so that's one important effect. The other, I think, is that when you do stuff like this at the ocean bottom, it's possible to keep dissolved gases dissolved very much longer. So here at the Earth's surface, if you, at atmospheric pressure rather, if you get too much hydrogen into the fluid, it will start to fizz out. Hydrogen doesn't dissolve very well in fluid. But as you get down to ocean depths and ocean pressures, you can dissolve a lot more, and that makes it possible for a lot more energy to be made available to the organisms down there. So the challenge is it's difficult to study these communities down on the ocean bottom because it requires typically access by submersible. So one of the best studied places actually is Lost City hydrothermal field, where there have been a few different expeditions, and will continue to be a little bit. But I think that we need to leverage the places that we can find right here at the surface. So here at the surface, you know, we can find a variety of different places that allow us to access these serpentinizing systems and go back there again and again and again. It's much easier to go and drill a hole, for example, a few hours from home in Northern California than it is out in the middle of the ocean bottom. So I think that working on both of those at the same time is going to really let us access a wider range of these places and really understand the full spectrum of serpentinization and what it means for life. Okay, another question from Sarah. What's your favorite thing about being an astrobiologist, and what are the perks? For sure, my favorite thing is that the questions that are addressed in astrobiology are such big and fundamental questions that it's just, you know, it's I mean, it's hard to imagine a bigger lure. And I think one of the maybe challenging things is that sometimes those fundamental questions, you know, what is life and are we alone and those sorts of things, can make the science a little bit or seem a little bit nebulous. And so the challenge is to find concrete things that you can investigate. And there are plenty of concrete things that allow you to walk a line between being specific, having, you know, a thing in front of you that you can actually study, and then taking those results and applying them to the bigger questions, to moving out and saying more general things about life. And so for me, one of the very interesting things is in studying the specifics of life, but in such a general way that those things are transferable to life beyond. So that's one of the things that I like the most about being an astrobiologist, you know, is the opportunity to work on questions that I think I really wouldn't be able to work on anywhere else. Another thing that I would say I really enjoy about astrobiology is that it seems like such a fascinating topic to the public that it's a real lure to draw people in public outreach venues and to engage them. So it gives you the hook that brings people in and then allows you to teach them some concrete things about science. And to have that possibility of capturing people's imaginations and then helping them learn something about that field is really important. And what are the perks? So one of the perks I've already described. I don't think there's any other thing that I could do, any other place that I could work, where I, as a chemist who got his start studying stinky ocean mud, would be able to help design future space missions. That's for sure a perk. And I would say another one of the things that I found very interesting is, or you know, a great thing for me, is that that work has taken me to some amazing places in the name of finding organisms or conditions that help inform our sense of life elsewhere. So I've been able to work on the ocean bottom and way up above the Arctic Circle and in Yellowstone Park and, you know, up in Northern California. I've just been to some really amazing places to pursue the sort of work that I'm doing. So, so for sure, that's a perk. Okay, another question from Adi. Do you have a favorite microbe or taxa, perhaps because of its unique metabolic activity, unique environment, symbiosis, etc.? You know, I got my start studying methanogens. So if you know, go for a paddle on a lake and you see bubbles coming up from down deep, chances are that's methane that's made by methanogens. A lot of us have methanogens as part of our normal gut flora. And so they're the ones that I understand the best. I find them interesting, and they're a lot of the basis for my study. But, you know, so that's sort of, you know, my academic interest. I guess the 10-year-old boy in me finds the organisms that literally transform the environment around them into acid as being very interesting. So you can find organisms that metabolize, that oxidize sulfur and iron, and literally create an environment of battery acid around them. And you know, if I were to ask my son that question, I'm sure those would be his favorite guys. And so, you know, so that little boy part of me kind of thinks those guys are cool too. Okay, another question from Julio. What are the fastest organisms we know of in terms of metabolism, and how fast could life go? So there, I guess I would have to look to what people have done in laboratory microbiology and the organisms that are best studied. So, for example, E. coli is a lab rat, meaning it's an organism that's studied very, very often, exactly because it grows fast. So E. coli doubles its population on the order of every 20 minutes or so. So that's an extremely fast organism. I don't know that it's necessarily going the fastest in terms of cell-specific metabolism, meaning, what's the metabolic rate of an individual cell, but in terms of growth, that's remarkably fast. And just to put that in context, the organisms that I mentioned earlier, way down deep in the ocean sediments that were going so slow, if you looked at their time of doubling, so how rapidly the population doubles, there at most, it's maybe once every few hundred to few thousand years or so. And here in the case of E. coli and some of its lab rat friends, that doubling is on the order of every 5 minutes or so. So I don't know what that is, maybe 5 or 6 orders of magnitude difference between them, which is a kind of an amazing dynamic range. Okay, a question from Lisa. What type of background do you recommend for someone who'd like to pursue a career in astrobiology? I think it really depends what interests you. I think that you can take almost any background in a more sort of traditional field in science and bring it to bear in astrobiology in a way that matters. So I think that you can be a physicist and find plenty of things to do in astrobiology. I think that's true of geology, of chemistry, of biology. So I think that having a background in any one of those things as a sort of, a strong training in traditional science is going to be important. But then, one of the things that I see in the people that are coming into the field now, so in the grad students and the postdocs and the young scientists, that really wasn't the case with a lot of the people that came in as my contemporaries, is there's a lot more overlap. So people have taken a lot more opportunity to think about the crossovers between physics and chemistry and biology and so forth. And so building that sort of thing in, maybe with a little bit of an undergrad research project or something along those lines, so that you start to have a sense of the connections between things, is important. And working in some kind of an environmental field or environmental question is a great way to do that, because I think there's no way to look at any part of the environment and understand it for what it is without bringing to bear multiple perspectives in science. And because, ultimately that's what astrobiology is. It's environmental science. It's just that the environments that we're thinking of are much farther afield and much more diverse than the ones that we have here on Earth. Okay, let's see. Any other questions? Any thoughts on looking for life on exoplanets in the follow the energy formalism? So, exoplanets are interesting and challenging all at the same time, because I think that there are just so many of them out there, that every time we discover a new exoplanet may be in the habitable zone, we sort of up our chances of having a place where life might exist. That's the great part. The bad part is, for a long, long time, the best that we're going to do is light through a telescope, a pixel or two worth of light that represents the entire planet, and maybe a spectrum that breaks that light down into a few, sort of chunks, so that we can get some very basic chemical information. But that's not nothing. There's actually quite a bit of information there. So how to do that within the sort of follow the energy formalism? I think that the way to do that really kind of goes back to what I said earlier, which is, life is one of the attributes of life is that it's capable of investing energy in a way that non-life can't do, right? So, for example, I mentioned that photosynthetic life here on Earth makes oxygen out of water, which requires a big investment of energy. And that also happens all the time in the atmosphere. So photons from the sun strike water molecules and make oxygen out of water. But life does it very much faster and very much more efficiently. I haven't done the calculation, but I'm guessing it's millions to 10 millions of times more efficiently on a per photon basis. So I think that finding a place where you see a signature, oxygen or something like that, that might or might not be life or based on life, that you can have abiotic and biotic mechanisms, but seeing it happen at a rate or with an abundance that it represents such a large investment of energy that there's just no possibility to see it as being not life. I think that's sort of the way to go. And that really means that, for me, I think when you look for the possibility for life in an extrasolar planet atmosphere, you're going to have to rely on, not a 1 or 0 answer, not that is absolutely uncategorically life or this is not. It's really going to be a question of degree, because the molecules that you're looking at have not very much information to them. If they can make it into the atmosphere in the first place, they're simple molecules, and chances are they can be made in other ways than life. So it becomes a question of degree. How much oxygen, or how much of a mix between oxygen and methane or nitrogen or something like that. And I think that's the sort of approach that should be pursued there. Question from Benu: what are some of your favorite books on astrobiology? So I will say, first of all, when I read, it's very often not in science, and it's very often not in science fiction. I think I sort of focus enough of my attention at work on science that when I read, I tend to read very different things. So I read a little bit more. Hermann Hesse is one of my favorite authors, and books that really don't have very much to do with science, maybe even more philosophy. But one of my favorite books that I don't think was ever written in the vein of astrobiology, but that I reference all the time, that I think is very relevant, was a book written by Erwin Schrödinger, the Nobel physicist. So he wrote a book called What is Life? And this was something that he wrote in 1944, which was prior to the discovery or the X-ray crystal structure of DNA. So he wrote this book in 1944, and it was based on a series of lectures that he gave at Trinity College in Dublin, which I wish I had been there for. And he wrote this book that is sort of a physicist's take on what life might be. And it's written in a very humble style. It's written in a very charming style. And yet here's this remarkable mind who's sort of turning to the question of what life is all about. And for me, it's interesting because it addresses life in the way that I myself try to do, which is to see life in terms of its general principles and not in terms of the specifics of the life that we see around us. So that's kind of an interesting one to pick up. It's very short. I don't think that you should worry that it's, because of being written by Schrödinger, that it's too complicated or deep. It's really not. He intends to write it for a more general audience, and so that I think is kind of an interesting one to pick up. Okay, question from Sarah. What are your thoughts on balancing research and outreach education as a scientist? Should individuals starting out focus more on research or try to do some of both? That's a question that's near and dear to my heart. So one of the things that I love about working here at NASA is that NASA understands the importance of education outreach. They understand that we are here, we do what we do, we have the privilege of doing what we do because it captures the public imagination. And so there's an emphasis on making sure that continues to happen and making sure that people are educated as to the science that we're doing in general. So to me, education outreach is extremely important. It revitalizes me in my job every time I have a chance to give a public talk or work at a museum or talk with school children or anything like that. It reminds me why I got into doing what I do, because it requires that I pull back from the day-to-day details of the science that I'm doing and re-articulate the big picture. And it's really important that happen. How to balance it? I think your credibility as in doing outreach really derives from your background as a researcher. You bring a different aspect to it when you are active in the field, and people can wrap their heads around that. So I think that it's really important to strike that balance. And for me, I guess I would say, maybe I spend 5% of my time, 10% of my time, something like that on outreach, probably closer to 5% or so. It's a 5% that really matters, but I think it is important to be a scientist first, and then an educator who educates by virtue of being a scientist. As far as early in a career, I think that it's probably different at most universities than it is at a place like NASA. So NASA, as I said, outreach is definitely encouraged. When you're at a university, whoops, I've lost you again. I'm going to have Sanjoy come back in and refresh. So when you're at a university, you have the demands of teaching, which is sort of its own form of outreach, although more formal, and your research program and all those sorts of things that you have to keep up with. And I mean, my recommendation is pursue the outreach as you're able and as your institution allows, for exactly the reason that I've suggested. I think it's a great way to refresh your interest in the field. Okay, one last question, it looks like, from Benu. How can we, as students and young scientists, use social media for outreach? That's I'm probably the wrong person to ask that question. I might be the only person in Silicon Valley who doesn't have a Facebook account. No, Craig doesn't either. That's two of us, I think, in the millions that live here. So, I think that it's actually a really important question because as a young scientist or a young person, I mean, you've really grown up with social media as context and have a much better facility with it than do even people of my generation. And I don't think of myself as old. It's something that came along after I already had developed my own habits. And so I would almost kind of turn the question around. I think that as students and young scientists, you understand the material, you understand the importance of outreach, you understand how to convey information to people, but you understand something that I don't, which is you understand social media and its use much better than I do. You know what's out there, you see what's emerging. And I think it's a really important job for new and upcoming people is to adopt that tool and use it in ways that I wouldn't know how to do. Okay, a couple more questions. One from Adi. Given the soil geochemistry results from Curiosity, is there a microbe we know on Earth that could live on Mars today or in the past? Simply if we added water, is there enough redox potential, abundance of elements, and are the only things missing pressure and temperature? That's kind of a good question. So almost everywhere that we go on Earth today, when we see microbiology there, we're seeing microbiology that's ultimately based on organic matter that's decomposing. And that organic matter originates with photosynthesis. So that's one thing that we really haven't seen yet on Mars. So we'd be relying on the chemistry. There are bits of chemistry that we've seen. So for example, gradients from oxidized to reduced species in sulfur and iron and things like that that have the potential to represent an energy supply. There are rocks that, when reacted with water, could liberate hydrogen or other species that represent energy. The question is whether that happens fast enough. So, as I mentioned, energy, is sort of everywhere. You look up on a starry night and you can see energy streaming down at you from the stars, but it's really not enough to run photosynthesis on. And likewise, chemical energy is everywhere, including in the sediments of Mars. It's just that we don't necessarily understand how much is required for life and whether it would be delivered fast enough there. So I think I have to give you an I don't know on that one. Okay, any final parting advice for students and postdocs interested in astrobiology? I'll just reiterate some of the stuff that I've said before, which is I think it's important to have a strong background in a traditional science. That you can then bring in and apply into whatever question you choose in astrobiology. So that's important, I think. I think that I probably don't need to say this to people tuned in or people interested in astrobiology, but it matters so much to have a field that draws you in terms of your intellectual interest and your fascination and so forth, because you get yourself a decade or so into a career, and it's important to still have something that keeps you excited about your work. And as I said, for me, the education outreach part of it, that kind of refreshes my interest. It reminds me why these questions are exciting and why, why I continue to pursue them. And so, it sounds a little bit silly, but I mean, really take the time to look around, figure out what gets you excited in terms of scientific questions, and then follow those pursuits. And I think that's about it. So I think that probably brings us to the end, I guess. It sounds like maybe there are no more questions. And I think maybe we're going to wrap it up there. So thank you all very much for tuning in. Thank you all for the great questions. There were some really good ones, and hope you had a good hour.